Now, if you look at cell cultures, historically, this culture of tissue sections from organs, this was established long back, almost in 1885 by William Rocks, when he isolated a section of the medullary plate of the chicken embryo and then he maintained it for several days in a warm saline solution. So this was one of the earliest examples of tissue cultures. This was followed by the American biologist Ross Granville Harrison, actually an embryologist, who in 1907 demonstrated the in vitro growth of neuronal fibers when a fragment from the frog's tadpole nerve cord cells were placed in a medium which contained the frog lymph. So this was one of the first ways established methods for in vitro propagation of nerve fibers, where the neuronal projections could actually be seen. Then in the 1910s, Albert Ebeling, he preserved the chick embryo from the cell line that was developed by Alexis Carrel. Alexis Carrel, as you can read this paper here that I have enclosed, he actually made several of these, and some of them became immortal cell strains. Okay, in fact, Carrel could subculture them for more than 19 times before he handed over to Ebeling, and Ebeling actually maintained them for 34 years by subculturing these cells repeatedly. And not only did he maintain them, he also used them as a model to test toxicity of germicides and many other experiments. In fact, this started the way for cell culture into various applications, and one of the biggest applications was the Nobel Prize that was given in 1954 to Anders Weller and Robbins for establishing a method for in vitro cultivation of the poliomyelitis virus in the absence of a nerve tissue. So this tells us the various applications that and the history with which the cell cultures have started. A cell line is what you normally use in the lab, and this cell line refers to a population of cells which are descended from a single cell, and they contain theoretically the same genetic makeup. These are supposedly also immortalized cells, and these immortalized cell lines, these are different from the primary cells which are derived directly from a subject, and therefore they have a finite lifespan. Now, when you isolate primary cells, we observe that they undergo senescence and they stop dividing after a certain number of population doublings, and this is called the Hayflick limit. On the other hand, immortalized cell lines, these are capable of proliferating indefinitely under in vitro conditions. Now, immortalized cell lines are not the same as stem cells. Stem cells also have the capacity to divide indefinitely, but they are part of the natural development of a multicellular organism. There are several methods for the generation of immortalized cell lines, and the most common one being the derivative of a natural cancer. Now, the oldest of such cell lines or immortalized cells are the HeLa cell line. These were derived from the cervical cancer cells from the patient Henrietta Lacks, who died in 1951, after whom the cell line, the He and La, is named from. August 2020 actually marks 100 years since the birth of Henrietta Lacks. But the story of Henrietta Lacks also illustrates the racial inequities that are embedded in US research and healthcare systems. Lacks was a black woman, and the hospital where her cells were collected, the Johns Hopkins Hospital in Baltimore, was one of the few that provided medical care to black people. It had taken samples of her cancerous cells while diagnosing and treating the disease. They gave some of the tissue to a researcher without Lacks' knowledge or consent. In the laboratory, her cells turned out to have an extraordinary capacity to survive and reproduce. They were, in essence, immortal. Now, the researchers shared them widely with other scientists, and they became a workhorse for biological research. None of the biotechnology or other companies that profited from her cells passed any money back to her family, and for decades after her death, doctors and scientists repeatedly failed to ask her family for consent as they revealed Lacks' name publicly, gave her medical records to the media, and even published her cells' genome online. Following an outcry, the genome was soon, of course, removed. Nature later published the genome of another HeLa cell line after the Lacks family reached an agreement with US NIH to approve its release. All this story about her and her cells, this is empathetically covered in the New York Times bestseller book, The Immortal Life of Henrietta Lacks by Rebecca Skloot. And in fact, this was also made into a movie, the same name of the movie too, and starring Oprah Winfrey. So you can watch that movie, or you can read this book. Okay, so what actually her descendants want is to just that the scientists acknowledge that HeLa cells actually came from an African American woman who has flesh and blood, who had a family and who had a story to tell. You can read more about her in this Nature article. Now, as we discussed, there are several cell lines that are commonly used in various labs. Here I'm giving you only some of the few major cell lines that are popularly used, but of course there's several cell lines that that are generally in use. And these are derived, some directly from the cancers and some in various methods. You can have spontaneous mutations. For example, that's how the mouse fibroblast cell line 3T3 were derived from. And you can also generate them by transformation. And one such cell line is a human embryonic kidney cell line, the HEK293. These are transformation of a normal kidney cells, but with the shared fragments of a adenovirus type 5 DNA. Okay. Now, if you look at this table, you can see 293T is one of the derivatives of these HEK293 cells only. Again, and both HEK293 as well as 293T cells, these are all widely used in cell culture because they have very high transfection efficiency. The 3T3 cells, which are derived from the spontaneous mutation, you can read about it. In fact, there's a nih3t3.com website for them. You can look at how these cells can be utilized for various research purposes. Then you have these cancer cells, A431, A549. These are skin cancer or lung cancer from, one is from an 85-year-old female, another is from a 58-year-old male. And some express EGFR receptors, some do not contain P53. Like that, there are various properties for each of these cells. C6 is again derived by tumors which are induced in regular astrocytes, which have been treated with N-nitroso-methylurea. Yes, so that's where you can derive, you can make a regular astrocyte cell line into a C6 glioma cells. Again, EGF receptors are lacking in CHO cells, which are derived from Chinese hamster ovary. And CHO cells are one of the widely used cells for industrial production of various recombinant proteins. COS cells, again, they are suitable for transfection, especially the SV40T antigen containing ones. And because that's how they are derived from by immortalization of the regular CV1 cells from the kidney of a monkey. HeLa cells we just discussed, and HeLa cells had actually been used for testing the polio vaccine, the SARS polio vaccine. These are the first human cells to have been successfully cloned, sequenced. They have been used in various cancer research, and in fact, to the extent that HeLa has, in fact, some people have called it to be altogether different species, in fact, because they have propagated for so many generations. HUVEC cells, since they are derived directly from the umbilical vein epithelium, this is a very good model if you are studying angiogenesis so that's in vitro conditions. There are cells like the IMR32, which although people use it as a neuronal cell line, but one has to remember that these are not derived from the neurons within the brain, but these are the neuroblastoma cells which are derived from a metastatic site, from the abdominal mass from where the neuronal cells have, or the early neuronal cells have undergone metastasis. So in fact, these IMR32 cells, these are actually a combination of two different cell types. Which are of course the neuroblast cells, which are predominant ones, but you also have several hyaline fibroblasts within the culture. Both of them together are actually propagating. So the results, one have to be cautious that these are not results that are coming only from a single or from a homogeneous cells. Jacquet cells, again, very common with immunologists and also cancer biologists, especially in HIV research and T cell signaling. So these again came from the patient suffering from leukemia. So in place of IMR32 cells, for a neuronal model, one can use Neuro2A cells, although of course IMR32 is a human cell human model, whereas Neuro2A is a mouse neuronal cell type. But these Neuro2A cells, these are derived from the albino mouse, okay, so in which contained a spontaneous tumor. Neuro2A have been typically used for routine diagnosis of rabies, so widely used in various research purposes, especially where viruses have to be grown in a neuronal cells. Then you have others, another common neuronal cells, which are the PC12 cells. Again, these are not typically neuronal cells, but these are cells deriving from the adrenal medulla. But they have the properties of the or show the properties of neuronal cells. In fact, they can undergo differentiation when they are treated with nerve growth factor, and they are typical model system for studying the synaptic vesicles. There are for the macrophages, one typically uses raw 264.7 cells, very common in at SAU also. These are typical cells used in various labs for macrophages as a model for macrophages. These have been derived from by inducing a tumor by the Abelson-Murin leukemia virus. So they are capable of antibody-dependent lysis of sheep erythrocytes and tumor cell targets, and so it's a very common model for using these cells in addition to inflammation studies. Then there are cancer cells, for example, directly derived from patient cancers. MCF7 is derived from a 69-year-old breast cancer patient, Frances Mallon, from whom we got the MCF7 cell lines. MCF, the word standing for Michigan Cancer Foundation, where it had been isolated and established. Our knowledge of breast cancer, in fact, comes much from this cell line. Then from a 62-year-old male patient who was suffering from prostate cancer, the PC3 cells had been derived. These are PSA negative and one of the highly metastatic potential among the cancer cells. Okay. They do not respond to androgens or EGF, and one can also create spontaneous tumors within the mice using these cells. SK and SH are again neuronal cells. These again are derived from metastatic site that is actually coming from the bone marrow in a 4-year-old female who was suffering from neuroblastoma. They of course exhibit a high level of dopamine beta hydroxylase. So this is one good cell line, especially when one is studying dopaminergic pathways. And it is also used in various cytotoxicity assays. So these are all, of course, a very short list of cell lines that I am presenting here, but there are various cell lines throughout the world. People use it according to their research purposes. You can search, or you can also purchase them commercially from a number of sources. A non-profit organization from where you can buy, and many of you are probably familiar with, is the ATCC, standing for American Type Culture Collection. So this is located in Virginia, USA, and they acquire. You can also deposit cell lines there. They acquire it, they authenticate it, and produce and preserve the cells, and they also distribute it. Okay. Not only just the cell lines, but including some bacterial strains and various media and other things, and there are also primary cells and stem cells too. Then there is another site, a non-profit one, which is established by the Public Health England, which established this European collection of authenticated cell cultures, ECACC. You can go to this website and you can buy things from there also. Commercial sources, Sigma Aldrich is a commercial source. Of course, they also sell the ECACC cell lines. Then there is a Coriell Institute for Medical Research, which is located in Camden, New Jersey. They have a large biorepository of cell lines. You can, if there are any cell lines that you are not able to find here, you can surely find there. And there are also various primary cells also. One can buy directly stem cells and other things from another source like Lonza, and especially media also with a very good media too. So these are various ways in which, and of course, scientists also often ask each other. That's another way through which one can get cell lines. In India, you have NCCS Pune, which also supplies many of these cell lines. Now, an essential requirement when working with cell cultures is to maintain aseptic conditions. Now, aseptic techniques, they essentially involve a number of precautions, and their aim is to basically, there are two aims actually. One is to prevent the cultured cells from undergoing contamination, and the second is that the lab worker also gets protected from against any possible infections. So for this, the most important thing is to have personal protective equipment, the PPE. It should always be used in a cell culture lab, and these include gloves, lab coats, eyewear if there is any protectiveness for eyes, then glasses are required. Otherwise, gloves and lab coats and covers for the shoes, these are absolute good biosafety measures that one should follow all the times. And if there are any sharp objects, needles, scalpel blades, glass pipettes, then one has to use sharp containers for separately, so that no kind of injury happens to oneself, and by that way infection can happen to you and contamination can happen to the cultures. Okay, so both can be avoided. One should use disposable plasticware, so that way one can avoid the puncturing of oneself with broken glass, for example. And of course, there is no need to autoclave these things. These are one can purchase them in sterile format, or one can autoclave them if you purchase in bulk. But of course, there is a high environmental cost footprint when you use disposable plasticware. For glassware and other things, sterilization one can do by autoclaving and followed by dry heat. Dry heat is generally 2 hours at 160 degrees centigrade. Despite all of these things, cell cultures frequently undergo contamination either with bacteria or fungus if any of these conditions are not met. And so it is very important that these are always there. Okay. Despite following all these aseptic conditions, one still sees contaminations. Okay. So that contamination can happen only if any of the aseptic conditions are not followed, because that is the only way through which bacteria or fungus can enter inside your cell cultures. So if you find it contamination, then most likely you have been certainly relaxed in one or other steps in the aseptic techniques. Now, why contaminations are dangerous? Because when you have contamination, then this bacteria or fungus, they inhibit cell growth because they compete with the nutrients that are present in the media, and your media is essentially nutrient-rich media. So it is a very favorite media for bacteria and fungus to grow in, very favorable for them. So if there is any relaxation in your aseptic maintenance, then these bacteria or fungus can easily grow there, and because they have a smaller doubling time, they will be growing at much faster rate compared to your cell line. And these bacteria and fungus, they also release toxic substances, which in addition to competing for nutrient sources, they will also release toxic substance which will kill your cell lines. And when the contaminations are very early and you don't catch it, then whatever results that you're getting from your experiments, they will be inconsistent, because these contaminations are going to change the metabolism of your cells. And if you have not seen the contamination and you continue to do RNA isolation or protein or whatever experiments that you're doing, then the results is not what will not be as expected or cannot be trusted to begin with. So therefore, it is very essential to maintain sterility of all the objects that are coming in touch with this, that you're going to put inside the biosafety cabinet. Okay. So the workbenches, the outer surface of all the plastic glasswares and the media bottles and everything has to be wiped with 70% ethanol and then only taken inside the cabinets. The water baths and the water tray that is kept inside the incubators, all of these have to be constantly changed and checked whether there is any fungal or bacterial growth happening there. The traffic in the room should be very low. Sometimes there are 20 people inside the cell culture room, which will increase the contamination issues. So the traffic should be very low. And cells should only be opened up within the biological safety cabinet, okay, where the airflow filters are essentially on, okay, and they are blowing out, okay. So there is no inward flow of air, but outward flow only. All the work surfaces, they have to be wiped with 70% ethanol or any other commercial disinfectant. You can read more about this at the CDC website that is enclosed here. There's a free booklet available there for you can download it, and it details all the aseptic techniques one can follow. For more details about PPE equipment, you can also check the website at Yale. For sterilization of all glassware and plasticware, we have already discussed autoclaving is essential, and autoclaving one does is at 120 degrees centigrade for 20 minutes at a pressure of 15 psi. If you're using disposable ones, you don't need to autoclave them, but then you have to open them only inside the biosafety cabinet. The biosafety cabinet, generally they come with a UV light for decontamination. But how effective this decontamination is, one has to, there is a lot of debate actually, whether it is really effective or not. Because many times people don't know whether the UV light that is coming is actually of good strength or not. Because the UV rays, they gradually, the glass tube that is transmitting the UV rays, they gradually lose the ability to transmit short UV rays over time. And then if the biosafety cabinet is cluttered, then many of the bacteria or spores which are not in the direct path of the UV, that is if they are present underneath the 6-well plate or underneath some tissue paper box or something, if they are present underneath or in any shadow region of any of these things, then these organisms can successfully thrive. So you can, so when you're turning on the UV light, the entire biosafety cabinet should be as empty as possible. And you also know if there is any flies or insects inside the biosafety cabinet, the UV light does not destroy it. So these insects, they can fly off once you turn on, once you open up the biosafety cabinet. But when they are inside, all the bacteria that are there under their belly and in the saliva, they are all very, very live. They are not destroyed by UV light at all. And once you turn on for your regular work, these bacteria are still around. Okay? So one should be careful that there are no insects inside the biosafety cabinet. Okay? So because of various these things, and of course there's a hazard for the person who is working. He can accidentally get exposed to UV if the door is open and the regulation between the door opening and UV light is not controlled. So for various reasons, people do not prefer or are not completely trusting on UV sterilization. So it is a secondary method of disinfection only. So primary method is the person has to be very careful in following all the other aseptic techniques. The lamps have to be periodically replaced. The UV lamps, they have to be periodically replaced, and annually they have to be tested for efficacy. Typically, one should use a UV flux meter directly under the UV light in the biosafety cabinet, and one should measure the radiation output. It should be less than 40 microwatt per square centimeter at 254 nanometer. Okay. Usually, one sees that this output is actually reduced with time. And if you are using a biosafety cabinet whose UV lamp has not been changed for like the past 5 years, then it is the same as actually not turning it on at all. Okay, because there is essentially no effectiveness in the whatever the radiation that is coming out. Sterilization of all work surfaces, one has to do with 70% ethanol. One can also use 10% bleach, or one can use quaternary ammonium compounds. Okay, so these quaternary ammonium compounds, which were first introduced in 1917, they are very good cationic surface active agents. These are generally used in 0.05 to 0.2% concentration, and these are active across a large pH from pH 3 to pH 10. So they interact with the bacteria in an electrostatic manner. The bacterial cell membranes or phospholipids, these are anionic, and then the quaternary ammonium compounds are cationic surfactants. So that's how they interact, and then they disrupt the cell membranes, leading to the cell leakage and then eventual lysis of the cells. Of course, QAC compounds are effective against gram-negative, less effective against gram-negative compared to gram-positive, and they are not effective against bacterial spores altogether or viruses. So for yeast and molds, one can use them, but at a higher concentration. Another source of contamination is the cell culture media. The media should always be sterile. If you are making your own media, then one has to autoclave it, and if any of the media components are heat sensitive, then you can filter sterilize it. The pore size for the filter should be around 0.2 microns. So anything less than 0.22 microns. An alternative method is to buy commercial media, which has already all the components added. So you can use those because they are already sterile, they have been packaged sterile, and they are basically sterile media only if you as long as you open it under the biosafety cabinet. In addition to following all these methods for aseptic techniques, one can have precautionary steps also, and these precautionary steps include like using antibiotic solutions in your culture media. Typical antibiotics to avoid bacterial contamination. So some antibacterial agents one uses are penicillin at a final concentration of 100 unit per ml, streptomycin 100 microgram per ml, gentamicin 50 microgram per ml, neomycin 50 microgram per ml. So these are some commonly used antibiotics. In vitrogen and many companies directly sell penicillin streptomycin combination, which is the most popular among the antibiotics. In few labs, people also use antimycotic solutions, especially if there is any person working with yeast nearby. Then it is very good to have antimycotic solutions also, because they will prevent the fungal contaminations. One such antibiotic solution is amphotericin B, which is used at 0.25 microgram per ml. Another is nystatin, used at 20 microgram per ml. Okay. In fact, there are commercially you can also buy a combination of penicillin, streptomycin and amphotericin B as a PSA combination, as an antibiotic antimycotic combination also. So these are various methods through which one can prevent contamination within the cell culture and maintain the aseptic conditions. A very important thing is that one has to look at the cells every day. When you are culturing the cells, you have to look at the cells every day. You have to check for three things. One, whether there is any cloudiness or turbidity on your culture media. If there is turbidity, that means there is some kind of contamination happening. Two, most media contain phenol red. We normally buy media with phenol red, which is an indicator for CO2, indicator for pH. If there is any contamination, media will turn into yellow color. So that again is your indication. Okay. So the media should always be in its original red color. Okay, that has to be always there. And the third is daily examination, microscopic examination of your cells. Okay, you have to you cannot grow the cells, plate the cells today, and because the passage is after 48 hours or after 72 hours, you simply forget about them. That should never be the case. Okay, every day one has to see, and one has to look for any abnormal morphology in your cells. Okay, if the cells have shrunk or they have become clumped, or any kind of extra growths or debris or fragmentations that you are seeing. So this one has to spend every day with your cells, at least 5-6 different areas within your plate one has to see. Okay, it is basically you're talking to your cells every day. That way one can avoid the possible contaminations within your cell cultures, and if there are any contaminations, one can early on itself one can address it if they are detected.